we're going to get started with the PyRosetta first lecture, which is going to be based on poses. And so to get started, we might want to think about working with certain biomolecular structures. So let's say we could have some type of a chemical compound. So we could have a drug, we could have RNA, we could have DNA, proteins, or these things combined. So what we want to think about is how we can sort of do data science, given that we can have different types of molecules. And so sort of trying to connect the concepts that we know and what Jeff has introduced earlier today with more code bases and sort of being able to compute or make calculations on biological data. What are some of the considerations? So what is something that we might consider before we get started? What should the pose class include? What is some relevant, perhaps, information or data about these biological molecules? Yeah. Maybe the size matters? Okay. Okay, there is possibly amino acids. Maybe there are some nucleic acids. So we might need to know. Okay, what else? What else does, let's talk about proteins. What else does a protein have in terms of maybe properties we care to compute things on? Yeah. Okay, so maybe chemical propensities, polarity, hydrophobicity. Yeah. Okay, so maybe we care about the side chain conformations. What else?
Maybe regions where it's a helix and regions where it's
Okay, so maybe we need to know the secondary structure, whether it's a beta sheet or an alpha helix, and have a way of keeping track of the three-dimensional structure. What else? Or can you see how we might use data perhaps related to structure? Okay, so a pose is the very basic object for PyRosetta. And so the idea of using a pose, let's say you have this protein with some zinc ions here, you want to be able to describe this molecular system and some of the properties that you've already mentioned. So structure would be a very important one. Obviously, this is a protein, so it would have an amino acid identity at each of the residues. And so you would need a way to store that information. And so there are different types of molecular systems. Once again, it could just be as simple as a drug or some chemical compound of that nature. It could be a protein and its ligand or ligands. So in this case, there are multiple ligands bound to this protein. Some proteins will bind RNA and DNA. So you would need a way to sort of store both the nucleic acids that belong to RNA or DNA, as well as its structural sort of coordinates. So exactly how to recapitulate the three-dimensional object. And we kind of brainstormed about what the class might contain overall, but try to put this, let's try to put it into more of the technical terms that we use. So given that we have the pose class representing, let's say, a protein, there's going to be a protein sequence. So a protein sequence, of course, contains the amino acid identities. And so we're going to have a protein sequence going N to C terminal. And we're going to have three-dimensional structure. So conformation is key to being able to assemble either a molecule or a macromolecular sort of entire assembly of a molecular system. And we might have some energy associated with maybe specific residues that correspond to binding site are going to have different chemical propensity, and so all of that is important, and we want to make physics-based calculations in PyRosetta and using PyRosetta. And so this, as well as other types of information in what is called a PDB file, is going to be key to what we do in PyRosetta. For those of you who have a favorite PDB and who have sort of spent a little bit of time taking a look at a structure, what is some information that we might have inside the file? So some examples, yeah. Okay, so we have coordinates, and that's at the atomic scale. So we have the atoms making up each residue and their coordinates. What else? Yep. Okay, so let's just say the sidechain atoms that belong to one residue identity. And so we have the correct number of sidechain atoms and listing their coordinates. Okay, what else?
I was gonna say like the domains, like
Okay, domains. Okay, we'll think about that in a second. Yeah.
the PDB file also contains extra information like how it's crystallized at the beginning.
Right. So we might have some experimental information regarding, for example, how it was crystallized or the resolution. So if you want to apply, let's say, a quality cutoff on the structures that we have, we would be able to do this based on the information stored in a PDB file. It will also have sort of the authors that deposited the structure. They will also sometimes have annotations, as well as they will name the protein based on most likely its function. So for de novo protein, the authors will say de novo designed blank. And so this, along with everything else we talked about pertaining to conformation and the atom identities, will all come together and be used for physics-based modeling. And so we need a way to organize this data and also store it so that we can efficiently perform certain computations. In terms of the sequences, here is a truncated protein sequence. So we're going once again from the N terminal to the C terminal here. And given a protein sequence, what we might need is its residue index. So we're going to have residue numbering, and the PDB file contains this data as well. And we're going to have a total residue count. So earlier in the introduction lecture, do you remember what the average protein length was that was mentioned? 300. So you would be able to gather that information from a PDB file as well. And then in here, we just have sort of one residue being shown. And we're going to see more in detail what it looks like once we load poses with PyRosetta. And we're going to have a residue class for specific information. So each residue has a set of atoms that make up the backbone and the side chains. And so we'll need to know the specific positions for each of those atoms. And from that, we can calculate the torsion angles. And you'll see that the pose class is very useful for this type of both angular and distance calculation. And so earlier we talked about domains. One thing to consider is you can have a PDB file that contains either a multi-domain protein or possibly a protein with multiple chain breaks. So raise your hand if you've seen an antibody structure before. Okay, so in antibodies, essentially you have the heavy chain and the light chain. So there are two different chains coming together to make an antibody. And so you can sort of think of, in order for us to calculate things, we need to be able to keep track of chain breaks. And we can do that with poses as well. And in terms of references to a residue type, so we'll be able to sort of both figure out chemical identities, and also whenever we want to design with Rosetta, we need to be able to pick certain residue types or perhaps a region, but more on that later. And so, as we mentioned, we'll need the residue name, some of the properties, as well as what coordinates, and something called its ideal coordinates. And so, quick question on, I guess, the difference between perhaps the coordinates that you get from the crystal structure from a PDB versus the ideal coordinates you could calculate for an individual residue. So what might be the difference between coordinates you obtain from the crystal versus idealized coordinates that you can calculate for a residue type? Yeah.
When you're trying to get the coordinates of a protein from the crystal, they're inherently kind of like vibrating because you can't just get them down to very low. So inherent that vibrating is going to cause a coordinate difference between ideal and the actual measured, and also there are flexible linkers and all that stuff that really depends on what is this maybe snapshot of time.
Yeah, so that's a very good answer, in that the crystal is a snapshot of what the protein structure might look like in ideal crystal conditions, but also you'll have flexibility in some regions, so this we can't really capture, let's say with X-ray, unless we specifically sort of have other experiments set up to calculate exact rigidity. And also, one other thing to consider is sometimes proteins are not going to have ideal coordinates to begin with. And so can you think of a reason that the crystal, as well as, for example, computationally you design a protein and it doesn't have the ideal coordinates? So maybe it doesn't have, it doesn't belong in the ideal torsion space. So what is just something that can sort of make perturbations to perhaps the backbone or the side chain coordinates? Yeah. Okay, so maybe if it's binding something and there's a little bit of strain in the 3D coordinates to accommodate for that function, then you can have some local changes to the ideal backbone. Is there anything else that could cause this?
Calculations, I think computation, some conditions, maybe the soul make up approximations of others, but these are not necessarily true in every context. So that could just cause some deviation from what you calculate than what is maybe captured.
Yeah, so we can have some, I guess, less than ideal constraints applied onto a protein structure and then make some perturbations that cause it to sort of look different than ideal. Okay, we can make changes to a pose in terms of its conformation, and then see how downstream from all the conformational changes that we have, what's the effect on the overall molecular system. And so this is something that we can both sort of track with the pose class and use it to make specific perturbations. Let's say we can take one specific residue position and change what the torsion angles are, and you'll get to do this in the exercise. We'll also be able to access and specifically read the pose geometry at any position. So this is a way to first inform yourself how to sort of get an idea of what it already looks like. I already shared the slides with you, but sometimes there are helpful snippets in terms of the code that you can use in your PyRosetta notebook. And so you can go back to some of the slides where it drops some hints, but the notebook will have hints such as this as well. Here are some examples of what it can sort of output when you look into the conformation. We might be interested in bond lengths and angles, as well as the torsions or other types of, let's say, sidechain specific, or maybe some calculation of interface. And so you'll be able to calculate all of this and make some changes today. And then we know in physics-based modeling, we talked about there being energy terms. So we try to calculate or approximate an energy such that we can have sort of guided computation. And so using PyRosetta, we might be interested in sampling, let's say, an energy landscape. So what you need for this is obviously some type of energies class, which your poses will contain some information on the residue level energies as well as sort of how it gets summed together. This is going to be an approximation. So do you think there will be a straight correlation to if you experimentally obtained energy-based values? So the answer is no. And tomorrow in the score function lecture, we'll explore why that is and what we can make of that. Some other information that we didn't talk about specifically is, so sometimes you have unique numbering of poses. In certain types of proteins, people work with their own numbering scheme. And they have different chain numbering scheme even. And so this can be complicated, and sometimes we want to standardize. So with the pose, with the PDB info, you can sort of read this type of information and make any necessary corrections. And you can also, what's not mentioned here, is you can read the header of the PDB file. So if you're interested in sort of reading the author section, you'd also be able to do that. And there should be a portion of that in the notebook. And so we're going to get started with manipulating the geometry of a pose where it contains a protein. And so we'll talk about phi and psi angles quite a bit today. And then let's see, so XYZ, what do you think that is? It's on the board. Yeah, so we have our coordinate system, and then we have the coordinates of atoms given our XYZ. And the other possibility shown here might be that you're interested in changing a residue to a new residue. So let's say you're interested in making a point mutation. So something to keep in mind here. And then we're able to create poses from scratch by giving some type of sequence. So in this case, you have 3 alanines here. And then use the ideal coordinates to initialize what 3 alanines sort of look like in a pose. And then you can make manipulations in terms of, let's say, phi, psi, distance, other XYZ, or even some mutation, and sort of get to your desired, in this case, very short peptide. The way the geometric changes are propagated in PyRosetta is not sort of based on, let me go backwards a little bit. So it's stored not in terms of your coordinate system and the XYZ coordinates for each of the atoms, but rather the bond distances and angles. And so you're making perturbations at the level of either XYZ or bond distances and angles, but these changes sort of come together and are stored in terms of those lengths, and also either the torsion or the dihedral angles or the planar angles. And so that is how you would be able to keep track of all these changes down the line. And we're not going to talk too much about fold tree, but there's a directionality to the changes you make in a pose. And if you're interested in this, some supplementary information on fold trees will be shared with you, but it is the material that got cut, basically, to make space for machine learning. And raise your hand if you're familiar with PyMOL. Should be everybody, because you had homework on this. It's okay if you just have the educational mode. You'll be able to do everything in these workshops with just that. But what you can do is you can visualize the changes you make to a pose, or you can take a PDB file and just visualize that in PyMOL. You should have already done that in the tutorial, but if you didn't, you'll get a chance to do it, and specifically with a pose. And You can also measure your structure by very convenient information. So this could be energy terms. So you could use PyRosetta to sort of store energy terms per residue and then visualize in PyMOL where it's the highest energy or the lowest energy. A question, let's say I have a protein that, let's go back to the very beginning. So let's say we have this protein structure and I, access all the information using the pose class, and then I calculate per residue energies. The question is, where would you expect the high energy residues versus the low energy residues to be? So this is basically the green structure is your protein, and you have some ligands bound here. And we're interested in sort of figuring out how the energy differs from, let's say, a certain residue that's placed somewhere versus another one that's maybe closer or further from something. So fill in the blanks, somebody.
Maybe residues on the inside is lower energy than the residues that's kind of like in water molecules.
Okay, so we're getting some terminology here in terms of perhaps the core of the protein versus the surface. So the residues that are pointing at the solvent are making different types of interaction. And so did you say the sort of exposed residues are higher or lower energy? Higher energy. And you would assume that the core is lower. Anybody agree? Raise your hand if you agree. Okay. We'll talk about it. Let's see, any other ideas besides core versus surface? Okay, so we're thinking maybe there's a difference in energy at the active sites versus, let's say, non-active site. And you would say it's higher at the binding site? Raise your hand if you agree. Okay, let's see, is there any other explanation you can think of for energy differences? Yes. Okay, very interesting. So one idea was perhaps these loop regions versus the very structured alpha helices and the beta sheets have different energies. And so which one do you think is higher versus lower?
Structures are lower.
Okay. Okay, that's interesting. So is there any other idea that somebody wants to just brainstorm? Let's see, so we covered sort of burial, so being at the core might have different energy terms, or it might just be slightly lower, possibly. We talked about there being an active site, and so that function changing the energy landscape, especially when the binding target is present, okay? And then we talked about there being a difference for the secondary structures that came together to make the protein at whatever position, and so that would have some trade-off. Is there any other idea that somebody can think of?
Around, like, are those metal ions?
Mm-hmm. Okay, so now we're talking about having energies for these ligands and that they would be different than a protein, a residue's energy versus, let's say, a free ion versus a bound ion. It's a little bit similar to the active site idea, but now from the perspective of what's actually being recognized, so specifically the ion. Let's see, is there anybody else? Okay, so I have two answers for you. the first one is, I never defined what the energy was, and you all answered anyway. But let's say we're making approximations to free energy, or we're trying to minimize based on some calculated pseudo energy. We will cover this extensively tomorrow. As for sort of everything else that was discussed and whether it's higher or lower energy, one thing that's great about this lab is you can investigate all of these different ideas and answer it yourself. So you can actually take a look at a core residue versus a surface residue and read out the energies and try to make some minor conclusions given, let's say, one protein. And you've all had some great points of discussion, so it'll be good to test these out. And with that, we're perfectly set up for the POSE lab. And once again, the slides are shared, so you can use the information that is shared